Hello friends, welcome to Bioninirmal. Today we will be talking about chromosome. Inside the chromosome there are presence of many genes, and these genes actually control the phenotypic character of many organisms. Of every organism's phenotypic character is actually controlled by the gene and which are located on the chromosome. Hofmeister first of all found some of the thread-like structure inside the plant cell nucleus Tradescantia edwardsiana. Suppose this is the plant cell and this is the nucleus, and Hofmeister seen some thread-like structure. So Hofmeister seen thread-like structure in the plant cell nucleus Tradescantia edwardsiana, and later Waldeyer gave its name as chromosome, and Waldeyer gave name chromosome. Okay, chromosome. Actually, chromosome means colored body. Chrome means colored and zome means body. So chromosome means colored body and zome means body. So chromosome means colored body. Each and every kinds of the organisms, and these are actually contain the chromosome. Suppose, and we will start with the virus. Clear? So this is the virus, and in the virus there are presence nucleic acid and either DNA or RNA. So the viral chromosome is either DNA or RNA, and this DNA may be double-stranded DNA and this may be single-stranded DNA, and RNA may be double-stranded and single-stranded. The RNA containing viruses are usually called as the ribovirus. So the viral chromosome is either DNA or RNA. In the prokaryotic cell, there are present a single circular double-stranded DNA molecule, and this is called nucleoid. So single circular double-stranded DNA molecule, this is called the nucleoid, and the nucleoid is the bacterial chromosome. Nucleoid is the bacterial chromosome. Apart from the nucleoid, there are presence of 1 to 20 small double-stranded DNA molecule. 1 to 20 small double-stranded DNA, and these are called plasmid. Okay, and this can be used in biotechnological process, and later we will see that how plasmid is utilized in biotechnology. But this plasmid actually help or protect this bacteria from various kinds of toxic materials, and bacterial chromosome nucleoid actually control many metabolic functions of the bacteria. But prokaryotic cells prokaryotic cells chromosome do not have any histone protein. Eukaryotic chromosome contain the histone protein, and this is the basic difference between prokaryotic and eukaryotic chromosome. Okay, and today I shall tell you about the detailed structure of the eukaryotic chromosome. Clear? And before going to investigate about the eukaryotic chromosome, first of all we should know that every kind of the cells in our body, there are present many number of the chromosome. Suppose this is the human cell, and in the human cell this is the nucleus. Okay, so this is nucleus, and inside the nucleus there are present chromosome, and these chromosomes are usually present in the two sets, and so these are the diploid, and so there are present 23 pairs of chromosome in the human cell. 23 pairs of chromosome, and this chromosome will be two types. One is autosome and sex chromosome. Clear? So autosome and sex chromosome. Autosome are 22 pairs. So 22 pairs are autosome, and one pair is sex chromosome. And this sex chromosome will be of two types. One is the X chromosome and another is the Y chromosome. Actually, genes are actually present in this chromosome, and this will control every characters of the human beings, and not only the human, every kind of the organism's phenotypic character are actually controlled by genes, and which are located on the chromosome. And later we will see the functions of the genes and how they control the phenotypic character. But now I am telling you about the chromosome structure. Okay. So the chromosome looks like the chromosome. So it actually looks like that, and there will be two arms of the chromosome. And here I can tell you that cell cycle. In the cell cycle, there are presence of the main two phase. One is the interphase, and in the interphase, this chromosome seen as the thread-like structure. So the detailed structure cannot be seen in the interphase stage, but in the M phase, the detailed structure can be seen because these are highly condensed. So highly condensed, and so the chromosome can be clearly visible. So this will be clearly visible. Okay. And here I am drawing you the M phase chromosome, and which is clearly visible. So in the chromosome there are presence of a particular constriction, and this is the primary constriction, and it is primary constriction a kind of the protein is associated, and this is the kinetochore protein, and this kinetochore protein contain many of the kinetochore microtubule. Okay, so this is the primary constriction, and these are the kinetochore proteins. So this is kinetochore protein, and these are kinetochore microtubule, okay. And so that in the primary constriction, the kinetochore proteins actually attached, and during the time of anaphase movement, this will be very crucial. These kinetochore proteins will be very crucial, and this kinetochore microtubule also will be very crucial. So this is the primary constriction, or this is called the centromere. Okay, so this is called as the centromere, or this is called primary constriction. Clear? And these are the two arms of the chromosome, and these two arms are actually called as chromatid. So two arms of the chromosome, and these arms may be equal or the unequal, and when this is unequal, then the short arm is called the P arm, and the long arm is called as that of the Q arm. Anyway, and inside the chromatid there are presents just these types of structure, and this is called as the chromonema. So this is the chromonema. So here is the chromonema. So this is chromonema, and in the chromonema some of the beaded structure can be seen, and this beaded structure, and these are called chromomere. So there are many beaded structure, and these are called as the chromomere. And when we will read about the nucleosome concept, then we will learn the detailed structure of this. So this is the chromomere, okay. And centromere, I have told you that centromere are the primary constriction, and this is the secondary constriction, and this is secondary constriction or another constriction in the chromosome, and this is called secondary constriction. And the secondary constriction actually contain 8Ts and 28S rRNA gene. So this is called, and this is responsible for synthesis of nucleolus. So this is called nucleolar organizing region. Okay, so secondary constriction is called nucleolar organizing region, and this is the structure of the chromosome. And the last part of the chromosome is the telomere. So this is the telomere. Clear? So in the chromosome there are presence of primary constriction, and just on the two sides of the primary constriction there are presence of the two arms of the chromosome, and this is called the chromatid, and inside the chromatid the chromonema present, and in the chromonema there are presence of some of the beaded structure, and this is called as that of the chromomere. And in the primary constriction kinetochore protein is actually attached, and kinetochore microtube will actually radiate from this kinetochore protein, and which will be crucial during the time of anaphase movement. Okay, and in the chromosome, the centromeric and the telomeric region will be very important. In the centromeric region, there are present highly repetitive DNA sequences. So there will be present highly repetitive DNA sequences. Highly repetitive DNA sequence. Okay, and in the telomere, there will be also present repetitive DNA sequences. So there will be also present highly repetitive DNA sequences. Repetitive DNA sequences. Okay, and this is actually the structure of the chromosome. And here I can tell you another important concept, that is the uniname and the multiname complex concept. So the concept is the uniname. So how many DNA? And here an important question can be arised, that how many DNA present in a chromosome? How many DNA present in a chromosome? Okay. So two concepts are actually given. One is the uniname concept and another is the multiname concept. So multiname concept. In the uniname concept, only one DNA in single chromosome. Only one DNA in single chromosome. And in the multiname concept, many DNA in single chromosome. Okay. So now it is discovered that this concept is the right. So in a particular chromosome, there are presence of single DNA, and with this DNA, the histone proteins will be associated. And how the DNA is actually packaged into the chromosome, or how the DNA is actually packaged into the nucleus, we will learn it about the nucleosome concept when we will read about the nucleosome concept. So this is actually the basic structure of the chromosome. In the next video, we will discuss about the chromatin structure, euchromatin and heterochromatin bar body, so everything. So if you like my videos, then you can subscribe my channels for more updates. So thank you guys. Thank you.